Hey class, let's get started with our second discussion for chapter 12. We're going to focus primarily on the second and third lines of defense. Our next chapter in your text focuses in great detail on the third line of defense, but let's kind of combine them together right now. Let's just briefly go over the first and second lines of defense. Before we can dive in and talk about that though, we need to define a term, the study of the immune system. Generally speaking, when we focus on immunology or the study of the immune system, we're going to focus on those second and third lines of defense. We don't spend much time focusing on that first line of defense. Generally speaking, the features of our first line of defense are included in other body systems. How we respond to those infectious agents that we're exposed to, and some autoimmune disorders or disorders of our immune system, such as allergies or cancer, can also be incorporated within the field of immunology. If we are working with an individual that has a very healthy, functioning immune system, their body is going to be surveyed. That immune system will look over the tissues of their body and patrol, whether it's using monocytes, whether it's using other phagocytes. It will survey the body for anything that doesn't belong, and if it finds something that it doesn't belong, it can recognize that substance or that pathogen, that microbe, as a microbe or substance that does not belong, and if necessary, it can destroy potentially harmful pathogens. A properly functioning immune system isn't going to destroy everything in our body that does not belong in our body. For example, if we eat food, we need to be able to differentiate food from a pathogen. So our immune system can tell the difference between a foreign material that does belong, or excuse me, a foreign material that is not harmful, and a foreign material that is harmful. To patrol our body, we're going to primarily use our white blood cells, or WBCs, also known as our I should probably spell that correctly, leukocytes. L-E-U, leukocytes, aka white blood cells. So we have lots of cells, these white blood cells patrolling our body. They are going to be able to recognize the body cells, the self cells. These are cells that belong in our bodies, and they can differentiate them from non-self cells. How do they do that? They look at the macromolecules on the surface. What kind of macromolecules? We're primarily going to focus on proteins and carbs. So depending on what proteins and carbohydrates are expressed on the surface of the cell, our white blood cells can recognize that cell as something that belongs in our body, or recognize that cell as something that does not belong in our body. When we start to mistake something that belongs in our body, a self cell, and mistakenly believe that it's a non-self cell, that's when we have an autoimmune disorder kick in. So if we mistakenly attack our own tissues or organs, it's oftentimes because our body's immune system is confused. It thinks something that belongs in our body does not belong in our body. So here's an excellent example. We can see here's a classic white blood cell. This classic white blood cell has receptors on its surface, and those receptors on the surface can have something fit into it, a surface marker protein or antigen. So here we can see a cell that belongs in our body. It is a self cell expressing self antigens, and when it binds to the white blood cell, there is no reaction. So woohoo, the white blood cell goes away. But then, our white blood cells can come into contact with the bacterium, and that bacterium will have surface marker proteins or surface marker antigens that do not match up with the receptors on the white blood cell. So instead of being a nice round receptor or a nice round marker fitting into a spherical receptor, it will call it a triangular bad marker that doesn't fit into the circular receptor, and that's going to trigger phagocytosis. That bacterium can then be phagocytized and then boom, 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 destroyed. Now, what do we look for on the cells, or what does our immune system look for? We look for those surface markers. Those surface markers on the cells that differentiate between self and non-self are going to be our proteins or our carbs, or aka sugars. They are going to allow our immune system cells to figure out what does and does not belong in our body. And if something doesn't belong in our body, generally speaking, we want to phagocytize that cell. We want to destroy that entire cell. If we can find a non-self protein that's not harmful for us, we should hopefully be able to recognize it as not harmful. I mentioned it briefly 2 slides ago. A good example of this is food. Food stuff that we bring into our body, so let's say that steak or that apple or that peanut butter and jelly sandwich, whatever it is you ate, it's going to have cells in it, plant and animal cells. And those plants and animal cells have surface marker proteins and sugars that we hopefully are not going to recognize as harmful, even though they are not self-markers. If we look at bacteria and other pathogens, there's typically some generic surface markers that are associated with generic categories of pathogens. These are called pathogen-associated molecular patterns, or PAMPs. PAMPs, a pathogen-associated molecular pattern. And these are going to help our body to recognize, generally speaking, that, oh, this is a bad bacterium, I should destroy it. Oh, this is a bad fungi, I should destroy it. These pathogen-associated molecular patterns are going to fit into pattern recognition receptors, PRRs. And these pattern recognition receptors are what the white blood cells use to identify self and non-self proteins and carbohydrates. So if we go back a couple slides here, let me go ahead and erase this. We have a receptor on the surface of the white blood cell. It's purple, and I drew it in red, but on the figure it's in purple. And that receptor is called a PRR, a pattern recognition receptor. Hey, see, it's even labeled down here. Woohoo. And this PRR is going to recognize the pathogen-associated molecular pattern, the PAMPs, that are present on the surface of this bacterium. And when a PAMP comes in contact with a PER, we initiate phagocytosis. That's right. We want a PAMP in the PER to get the phagocytosis. And yes, I enjoyed saying PAMP and PER. So, moving on. If our immune system recognizes a pathogen-associated molecular pattern with a with our PRR, our pattern recognition receptor, will trigger phagocytosis. Our immune system, as it's going about its business of patrolling our body's tissues, is not going to be localized in one site. By definition, it can't be localized in one site. As it's patrolling our body's tissues, our immune system is going to be distributed throughout our body's tissues. So our immune system is very diffuse. It's diffused throughout the cells, throughout the tissues and organs of our bodies. So we have a little bit of our immune system in all of our organs and tissues. Because it's diffused throughout our body, this helps us with surveying our body's tissues for any potential harmful substances and or pathogens. Once something is identified in our body that doesn't belong in our body, we need to have a coordinated, effective response so that our immune system can eliminate it as efficiently as possible. On that end, we typically are going to have problems because our body is compartmentalized. For those of you who've taken human anatomy, you've learned about body cavities. There's a thoracic cavity with the mediastinum, the pericardial cavity, the pleural cavities. We have the abdominal pelvic cavity. We have the retroperitoneal cavity. These different cavities of our bodies have double-membraned serous membranes that help separate them from other membranes or cavities of the body. And it's tough to get something from one cavity to another cavity across the membrane, to have that communication across the membrane. So how do we do this? What do we use to compensate for that? We use the mononuclear phagocyte system, the MPS. There's going to be a lot of acronyms in this chapter and the next chapter. Mono means one, nuclear refers to nucleus, phago means eat, cyte means cell. So this is a one nucleus eating cell. In other words, this is going to be a big cell with one nucleus that eats other cells. These mononuclear phagocytes in the mononuclear phagocyte system are in direct contact with tissue cells and are in direct contact with the extracellular fluid. They're floating around in the extracellular fluid. Within the compartments that are in contact with the extracellular fluid, so all compartments of our body, because extracellular fluid is everywhere in our bodies, we are going to have blood vessels and blood capillaries and lymphatic capillaries penetrating into these compartments. So these mononuclear phagocytes are going to use the bloodstream to rapidly move from compartment to compartment in our body. The chemicals that originate within the mononuclear phagocyte system and the extracellular fluid are going to go from the mononuclear phagocytes, diffuse through the extracellular fluid, and then diffuse into the blood capillaries in the bloodstream and the lymphatic capillaries, and then trigger the system-wide alert. Here's a diagram for your text showing that we have relatively higher concentrations of our phagocytes in the thoracic region. We also have them in the hepatic and the gastric region, and the inguinal region, and to a lesser degree the popliteal, the cubital fossa regions, and our ankles as well. Now, as our mononuclear phagocytes, or one-nucleus eating cells, are moving around throughout the tissues of our body, there needs to be a little bit of space in between the cells of the tissues of our body for those mononuclear phagocytes to move through. Holding the cells slightly apart, we have reticular fibers. And those reticular fibers are what the mononuclear phagocytes move across as they move between cells of our tissues. Here we can see we have a neutrophil, we have a macrophage, and we have a dendritic cell. Neutrophils, macrophages, and dendritic cells are all examples of mononuclear phagocytes. Mononuclear phagocyte is a broad category of one nucleus phagocytotically active cells. This reticulum is made of the reticular fibers. It has those reticular fibers and other connective tissues that permeate, connective fibers that permeate all the tissues of the body, and they connect nearby cells with each other so that we have a way to hold our cells within the tissues in place and hold the organs in place in our body. This reticulum is intrinsic to our immune function because it allows for those mononuclear phagocytes to move between tissues and organs. We're going to find lots of those phagocytotically active cells in the reticulum. There are high concentrations of reticulum in these following spots in our body. We have the thymus, which is located right around the larynx, right above where the primary bronchi originate. And we have lots of T lymphocytes that mature in the thymus. We'll also have lots of mononuclear or phagocytotically active cells within our lymph nodes, tonsils, spleen, and other lymph node tissues of the gut and respiratory tract. The mononuclear phagocyte system is loaded with white blood cells. These one white blood cell that we haven't mentioned yet in the mononuclear phagocyte system is a macrophage. A macrophage literally means big eater, the big eat, so to speak. So a macrophage is a really big cell that patrols our body and will phagocytize other pathogens that do not belong in our body. These macrophages wait and will attack through phagocytosis any foreign intruders that they come in contact with as the macrophages are waiting within those respective tissues. If we look at our lymphatic system in general, the lymphatic system is a subset or a subcategory of the immune system. The immune system and lymphatic system go hand in hand with each other. You could think of the lymphatic system as the other circulatory system in the body. When most people think of circulation in the body, they think of the cardiovascular system. The blood getting pumped to the heart, getting pumped through arteries and veins, back to the heart, so on and so forth. But we have a secondary circulatory system in our body. The secondary circulatory system is the lymphatic system, and we'll have separate vessels and specialized accessory organs that will transport lymphatic fluid or lymph through a series of smaller to increasingly larger vessels. Eventually, all of this lymphatic fluid that's been transported through the lymphatic system drains back into the circulatory system near the large veins that are next to the heart. So what's the lymphatic system do? Generally speaking, the lymphatic system regulates fluid balance in our bodies. So as liquid is leaking out of the leaky capillaries in our body, we are going to have that extracellular fluid, the excess extracellular fluid, get sucked into the lymphatic system and drained back into the cardiovascular system. We also use our lymphatic system as a drain-off to help remove inflammation from the inflammatory response. So if you have a lot of swelling or tumor because of an inflammatory response, that excess liquid that's causing the swelling will be removed from the appendage or the region of swelling via the lymphatic system. Our lymphatic system is also going to survey the extracellular fluid. That extracellular fluid, as it's sucked into the lymphatic system, is going to pull with it any foreign materials that may be in the extracellular space. And those foreign materials are going to be filtered and concentrated in areas of our lymphatic system that have high concentrations of lymphocytes, phagocytes, and other antibodies. So what is this lymphatic fluid? Lymphatic fluid is very similar in composition to extracellular fluid. If we look at the extracellular fluid and the lymphatic fluid, they're almost identical to each other. The big difference between them is the location. It's all about the location. If it's located in just the extracellular space, it's extracellular fluid. But the moment that extracellular fluid gets sucked into a lymphatic capillary, it is now recategorized as lymphatic fluid, or lymph. It's chemically nearly identical. It's just in a different location. Lymphatic fluid forms originally when we have liquid leak out of the blood vessels. That liquid that leaks out of the blood vessels is called extracellular fluid. Within our lymphatic fluid, we are going to have lots and lots of white blood cells, WBCs. These leukocytes, these white blood cells, use the lymphatic system as a secondary transport network. They have an alternative way to move around the body. And as this extracellular fluid is getting sucked up into the lymphatic system and being recategorized as lymphatic fluid, we're also going to have lots of cellular debris being moved with it. We're going to have infectious diseases or agents being moved with it. And if we look at the lacteals within the microvilli of the small intestine, we also use lymphatic fluid to transport lipids that we're in the process of digesting and absorbing. These lymphatic vessels that move the lymphatic fluid of our lymphatic system are going to be very similar in composition to thin-walled veins. They are going to have a very thin tunica media, so there's a very, very thin layer of smooth muscle associated with the lymphatic vessels that's effectively negligible. It effectively doesn't do anything. Our lymphatic vessels are going to depend primarily on the contraction of skeletal muscles to move the lymphatic fluid through the lymphatic vessels, just like veins depend primarily on the contraction of skeletal muscles to move blood through the veins back up to the heart. Our lymphatic vessels are going to be accompanying blood capillaries. Why do lymphatic vessels accompany blood capillaries? I'll tell you why. Blood capillaries are very leaky. And as these leaky blood capillaries are giving off lots and lots of extracellular fluid, we need to suck up the excess or the extracellular fluid into lymphatic vessels. Since we have blood capillaries in all parts of the body, we're going to have lymphatic vessels in nearly all parts of the body as well. There are a few notable exceptions. In our nervous system, within the central nervous system, we don't have lymphatic vessels. Instead, we'll have cerebral spinal fluid being circulated as opposed to extracellular fluid. If we look at other organs like bone, particularly bone tissue that is compact or compact bone tissue, there's no room for capillaries in the compact bone tissue. It's a solid mass of crystallized minerals. So we don't have any excess fluid, therefore we don't have lymphatic vessels there. The placenta is actually part of a separate organism. It's the child, not the host parent. And then the thymus itself also doesn't have any lymphatic vessels. Our thymus is one of the few organs of the body that has a high blood supply, but also does not have lymphatic vessels. These lymphatic vessel walls are very easily permeated by extracellular fluid. That makes it easy for them to absorb that excess extracellular fluid. And the lymph that's absorbed into the lymphatic vessels will, generally speaking, move towards the heart. The general direction of travel is always going to be towards the heart. Within our lymphatic vessels, we'll find that there's very high concentrations of them in the hands, feet, and around the areola of the breasts. And let's talk about a neat little tangent. I remember when I was younger, I always wondered, why is breast cancer so dangerous? I just didn't understand it. It's not like having breasts was an essential life function. It's not like a brain or a liver or a heart that you absolutely need to stay alive. Why can't you just cut off the breast and remove the cancer? It's not necessarily the cancer in the breast that's the problem with breast cancer. The problem with breast cancer is the high number of lymphatic vessels within the breasts that make it super duper easy for breast cancerous cells from the breast cancer tumor to migrate to other parts of the body very rapidly. And that's why breast cancer is so dangerous. So here we can see our two circulatory systems in our body. We have the cardiovascular circulatory system, we have our lymphatic circulatory system, and they can be superimposed on each other. Our lymphatic system drains back into the cardiovascular system near the large veins of the heart. Take human anatomy, we'll talk about those in more detail. Let's focus on our thymus. That thymus is one of the primary organs of our immune system. Why is it a primary organ of the immune system? Because it is responsible for making our T cells. When you see the term T cell, I want you to think of thymus cells. A T cell is also known as a T lymphocyte. So this term, T cell, is equivalent to T lymphocyte. Our thymus is going to originate when we're in utero, like most of the organs of our bodies. It starts forming before we are born. It's located at the inferior end of our trachea, right before the primary bronchi branch off of the trachea. These thymus cells are going to develop specificity. Particularly the T cells are going to develop specificity. So they are going to recognize very specific antigens. And after they develop that specificity, they're released into the cardiovascular circulation as mature T cells, as opposed to going through lymphatic capillaries. After those T cells are released into the cardiovascular system, they are then going to migrate and settle at lymph nodes throughout the body and in the spleen. Lymph nodes, now that we're talking about lymph nodes, are small regions of the body where we have lots of lymphatic vessels concentrated and merging with each other. You can think of them as a relay station, so to speak. They're small, about the size of a large kidney bean, and are going to be located in clusters along our lymphatic vessels, or channels in our body. We have high numbers of lymph nodes in our thoracic and abdominal pelvic cavities. We'll also have lots of lymph nodes in the axillary region, the inguinal region, and the cervical region of our bodies. If we are actively fighting an infection, these lymph nodes are going to swell up. So if you go to a doctor's office and the physician or healthcare provider palpates your neck to feel for swollen lymph nodes, they are checking to see whether or not your body is actively fighting an infection that would be indicative of swelling of the lymph nodes. Every once in a while there's an infection that doesn't cause the lymph nodes to swell, but the vast majority of infections do cause swelling of the lymph nodes. Another primary lymphatic organ is the spleen. Our spleen is kind of a jack of all trades. It does multiple things. It's hard to peg it and put it into one organ system. We find the spleen in the upper left quadrant of our body. It's right next to the stomach, kind of close to the pancreas, and it's right next to the splenic flexure of our large intestine. So it's actually the namesake of the splenic flexure. I digress. So our spleen is going to filter blood instead of lymph. You can think of our spleen as a blood filter. And unlike the kidneys, which also filter blood, our spleen is primarily focused on removing pathogens from the blood and removing worn out red blood cells. Our kidneys are primarily concerned with filtering out dissolved toxic chemicals from our bloodstream, the soluble toxic chemicals. Our spleen filters out the solid, unsoluble materials from our bloodstream. Why do we need to remove the worn out red blood cells? We remove and process worn out red blood cells in our spleen because we need to recycle the iron from the hemoglobin present in those red blood cells. If we have loose iron or unbound iron floating around in our body, that's going to make it very easy for bacteria to grow. By minimizing the amount of extra or excess iron floating around in our body, we can minimize the amount of bacterial growth, and our spleen aids us with that process. Adults that have had their spleen removed, generally speaking, are going to be okay. You know, the spleen helps us with our immune system, but as an adult, we've already established our active immunities, and there aren't many more pathogens that we haven't been exposed to yet. So we're going to be okay. Our teeth, we've established a good repertoire of memory T cells in our bodies. But young children and infants who have had their spleen removed are going to be severely immunocompromised. By removing that spleen early on in their lifetime, you make it very difficult for them to establish active immunity with their T lymphocytes to have those T memory cells build up. A secondary function of the spleen is that it's a blood reserve. It's a storehouse for extra blood. So if you are ever in a situation where you're bleeding a lot or having lots of hemorrhaging, you are going to release extra blood from your spleen to hopefully stave off or avoid sanguination. We also have some other lymphatic tissues in our body, but these are more diffuse and harder to pin down. All the tissues on this slide are generally speaking referred to as secondary lymphatic tissues because we can get rid of a good amount of them and still be okay. We have gut-associated lymphatic tissue, aka GALT. Our GALT, gut-associated lymphatic tissues, are going to be spread out primarily through the digestive tract. These gut-associated lymphatic tissues are discrete bundles of lymphocytes that are just beneath the surface or at the surface of our intestinal mucosa. Why do we want to have lymphocytes monitoring what's flowing through our intestines? Well, we want that to happen because we ingest many pathogens. So we need to sample what's getting brought into our body through our mouths, and this GALT, this gut-associated lymphatic tissue, is kind of like a first responder, so to speak, in that it gives us an early warning for any pathogens that may be potentially entering into our bodies. It gives us that first strike potential against that constant influx of microbes that we're consuming. We have lots of GALT in the appendix, within the lacteals of the microvilli, and within their Pryor's patches. Pryor's patches are a specific kind of GALT. So, just to make it clear, the appendix is not a vestigial organ. It serves a purpose. A vestigial organ is an organ that we do not know the purpose of yet. We now know that the appendix aids us with our immune function. Now, because this is a secondary lymphatic tissue, we can live on, we can be okay if we lose a little bit of it. But if we lose our appendix, if we lose our tonsils, which are other secondary lymphatic tissues, and if we lose the spleen, that's a lot of secondary lymphatic tissue. But if we remove high quantities of secondary lymphatic tissue from our body, then you will be immunocompromised. But just removing the appendix all by itself isn't going to impair the immune system of a patient. We're also going to have sites that are similar to the GALT, the gut-associated lymphatic tissue, in the respiratory tract, in the tonsils. And these are going to have very similar purposes to the GALT of the digestive tract. If a woman is pregnant and or lactating, she's also going to have temporary sites of antibody producing within her lymphoid tissues of her breast. And those antibodies are being pumped into the breast milk, the colostrum, the foremilk, the hind milk, so on and so forth, to help give baby a good bump and the baby's immune system, give the baby some passive immunity from mom. Let's look at blood. Blood in the cardiovascular system is going to be a way that we can move white blood cells and chemical messengers throughout our body to help different aspects of our immune system communicate with each other. If we look at whole blood or unaltered blood, we're going to have blood cells, aka formed elements, and those can be broken up into the red, with an R, red blood cells, white blood cells, and the platelets, aka the erythrocytes, the leukocytes, and the thrombocytes, respectively. We also are going to have plasma in our blood. This is the liquid portion of the blood that contains dissolved elements of our blood. It's clear and has a yellowish tint to it. That yellowish tint to the blood plasma is from all the dissolved chemicals in the blood plasma. And we also have serum. A serum, for all intents and purposes, is effectively the same thing as blood plasma. A distinction between serum and blood plasma is that serum is going to be the liquid that we extract from clotted blood. So if we trigger clotting in the blood, and then suck the liquid or remove the liquid from clotted blood, that is serum. If we remove the liquid from unclotted blood, we call that plasma. We can use serum in immune system testing and in immunotherapy. A generic term for making blood cells is hematopoiesis. An old school term for this was hematogenesis. but most texts now are shifting towards the term hematopoiesis. So hematopoiesis is the generic production of blood cells, and then there's subcategories underneath this umbrella term, like erythropoiesis, leukopoiesis, thrombopoiesis, to make the red, white blood cells and then the platelets. But hematopoiesis is the umbrella term that incorporates production of any kind of blood cell. Now, any kind of blood cell is going to need a precursor cell, regardless of what cell type it is. Our precursor cells in our body that differentiate into specific cell types are called stem cells. If you take human physiology with me, we spend a lot of time talking about totipotent, pluripotent, multipotent stem cells, but that's not what we're going to talk about today. Just know that we have a slightly specialized, it's kind of specialized to be a precursor for blood. During development, these stem cells go from being very general, undifferentiated, to being partially differentiated, and then when they go from being partially differentiated to fully differentiated. When they become fully differentiated, they're the red blood cell, they are the white blood cell, or they're the platelet. A white blood cell, a WBC, aka a leukocyte, is going to be programmed to turn into several different kinds of white blood cells. There are multiple kinds of white blood cells in our body, then they each have different functions within our immune system. Now, if we look here, we can see that we have our precursor hematopoietic stem cell. So this is, for those of you who are curious, this is a multipotent or adult stem cell, and it's been partially differentiated to let it know that it can turn into any other kind of blood cell. We have red blood cells, that's the vast majority of our blood. We also have our platelets as well. We are not going to spend a lot of time talking about red blood cells and platelets with the immune system. We're focusing primarily on differentiated white blood cells. And of those differentiated white blood cells, these guys over here that I'm circling in blue are part of our secondary immune response. So these are generalist cells. So the neutrophil, basophil, eosinophil, and monocyte, they all have generalist response. The monocyte can turn into a macrophage or a dendritic cell. If we look at this myoblast, the myoblast can turn directly into a mast cell. So in blue, circled in blue, we have cells of the secondary immune system, or that are responsible for the secondary response. And then over here, that I'm circling in red, we have our lymphocytes. So the precursor lymphoblast will turn into the B cell or the T cell. Ooh. This natural killer cell. That natural killer cell is actually part of the secondary immune response. I didn't mean to circle that one in red. But we'll have our lymphocytes, our B cells and our T cells, that are primarily responsible for the tertiary or third line of defense. So, class, concept check. Lymphatic fluid contains all the following except white blood cells, extracellular fluid, red blood cells, cellular debris, or infectious agents. You can rewind the video to look up the answer, flip back in your textbook, or flip back in your notes. Pause this video and get me an answer. 1, 2, 3, 4, 5. Okie dokie. Lymphatic fluid is used to transport white blood cells, so of course it's going to contain white blood cells. Lymphatic fluid can also be used to filter out infectious agents, and if it can filter out infectious agents, it must have the infectious agents in it. Lymphatic fluid also has cellular debris in it as cells break down, and it is formed from excess extracellular fluid. The lymphatic fluid should never have red blood cells in it. The red blood cells should only be in the blood within the cardiovascular system and should never get into the lymphatic vessels or the lymphatic fluid. So, if you have any questions, please feel free to shoot me an email or post them on the discussion board. As always, I'm here to give you guys a hand. Happy studies.